Hello everyone, and welcome back to our next lecture over metabolism for our asynchronous microbiology course. So this lecture is going to cover metabolism, what metabolism is, kind of the things that are involved in metabolism, and a little brief overview of kind of how metabolism works. So, metabolism is usually referred to as kind of the sum of all chemical reactions inside of a cell. So this is going to be cells making energy, cells breaking things down to get the energy stored in them to use to make energy later, and things like that. So all the different chemical reactions inside of a cell can basically be broken down into two different forms. You have those that break things down to get the basic raw products, the building blocks, if you like, out of them, or the energy that's stored inside of them out. And that's called catabolism. So breaking things down, catabolism, breaking things down. And then you got the opposite of that, which is building things up. So this is going to be taking those small building blocks and using them to make larger things like proteins or DNA, things like that, ATP from them. And this requires energy to be put into it. So catabolism, you're breaking things down, you're getting the energy that's stored in and out. In anabolism, building things up, it takes energy to do so. Pretty easy to figure that one out. So, onwards. Now, inside of a cell, we need things to do work, to break those things down, break nutrients, things that the cell brings in. We need things to break those things down, and we also need workers, if you like, to build things up for us, to can put different chemicals together, stick different sugars together, stick different ATPs and things like our different phosphate groups and things on ATP. And we need things to do that for us inside of a cell. It doesn't just magically happen. So the workers, if you like, inside of a cell are things called enzymes. And you've heard everything about enzymes before. It's nothing new. They make reactions, chemical reactions occur that either would take a lot of energy for the cell to start initially. They lower that energy for us. Or they make things happen faster than a cell normally could do so just by itself. So both of those things are nice. Enzymes, for the most part, they lower things called activation energy. It's kind of the amount of energy that it takes to start the ball rolling down the hill. If you have a car jack and you can kind of crank the rock up and get it rolling for you without you just kind of push it, or somebody else helping you push, that lowers the activation energy, if you like, that the cell would have to put in to start that chemical reaction. So enzymes, they work by kind of making things closer together. So instead of the cell having to go find this one little molecule and this one over here to stick them together, the enzyme will kind of wait for one to float into it, and it waits for another one to float into it. And then things are close together. So now it's just a little bit of energy to stick them together. Instead of all the energy, you have to go find that one and find that one and track them down and stick them together and things like that. So enzymes kind of help with that. They can also make, like I said, they can make things go quicker. But the nice things about enzymes is they don't get tired. They don't degrade over time with use or anything like that. And once it does something, it makes a change, it puts two things together or breaks things apart. It's not going to be changed at all. That enzyme can go right back to doing that again. So enzymes are indefinitely reusable, which is super cool. So unless the enzyme just gets damaged or something like that, which we'll talk about in a little bit, cells can use those enzymes pretty much for once it's created for the rest of that cell's life, kind of thing, which is super cool. Like I mentioned, they make things faster. They make things easier to do by pulling things closer together that the cell doesn't have to go find, and they make them closer together, kind of thing. So you've heard this one before, the structures of enzymes. There's two different forms of enzymes. We have something called a conjugated or a holeoenzyme, and then just a simple enzyme. And you can kind of think about this like words. A conjugated word is two words stuck together, like don't or won't or something like that. Will not, do not. So two enzymes stuck together. And then you have a simple enzyme, which is just a simple word by itself, which is literally just a singular protein by itself just floating around, and that's the enzyme that does something that it needs to do. A conjugated enzyme is where those concepts of micronutrients and things are zincs and cofactors and enzymes and things like that come back into play. And this is what those are going to do. So our conjugated enzyme has the protein part, the thing that's actually going to do something and move things around. And then you have the non-protein part, which is going to be considered the cofactor. And that is going to kind of be the kind of like you don't necessarily need you have to put the lock, you have to twist the lock and put the key in at the same time. Without the key, you can't twist the lock, and that's kind of the same way that this works. So a simple enzyme is literally just a lock that doesn't have a key. You just twist it and open. Whereas a conjugated enzyme, you have to put the key in first, twist that before you can twist the lock. And that's what the difference is. So simple enzymes don't have locks, you just go right on in. Whereas our conjugated enzyme has a lock, you have to have the key in first, which is the coenzyme, to make it work. And that's why getting those little trace elements and things are so important in our diets, and also why we have to provide them to bacteria, because without that little key, they can't open the door, their enzymes won't work, so they can't do what they need to do. So here's a couple of the common enzymes and what they do. Take a look at those if you wish. So I'm sure you guys have heard about 1,000,000 times over about the concept of protein folding and things like that. Well, the protein folding and the way that enzymes are folded is what makes them specific, and that kind of stuff. So you've probably heard this over and over and over again. I'll post a review video if you need it. So the induced fit and things, you probably heard this one before. Cofactors and things you probably or may not have heard before, but they help the enzyme work. Once again, they're a lot like that key and lock concept. Simple simple enzymes that don't need a cofactor to work. You just essentially twist the door lock and go on through. Very simple. They don't require these cofactors and things to work. Whereas oleoenzymes require an extra little step in there. It's that lock. They require the key before you can open the door, kind of thing. So what these are going to do is, you can kind of see here that this particular little substrate that's going to be fused, substrate 2 that's going to be fused to substrate 1, they don't necessarily fit really well together. There's a little blank space here that's missing. So what that coenzyme is going to do is it's going to kind of fill in that extra space. It's going to, even though they don't necessarily fit, it's going to kind of make sure that they fit. Or what it might do is it's going to take, in this case, it's going to take this little red ball here from substrate 1, and it's going to move it over to substrate 2. So you can see here it moves it over and bonds it to substrate 2. So these two things don't ever actually touch, and that coenzyme is going to transfer some chemical group or things like that. So coenzymes do lots of different things. They just help enzymes do things that they might not necessarily be able to do if it was just a protein structure. So it's pretty cool on what these guys do. Now there's two different types of enzymes when it comes to where they're used inside of a cell or outside of a cell, and that's exactly what they are. So the vast majority of enzymes that you find on the planet are going to be produced and used on the inside of a cell. So the cell is going to produce them to, make new DNA, make RNA polymerases and things like that. Or it's going to produce them and get them out of its cell. It's going to need them to digest things. So that's an exoenzyme. So an endoenzyme is going to be the vast majority of them, the ones that cells need to maintain their life as a cell. Things that are involved in metabolism, things that are involved in making new DNA and RNA and things like that. And they're going to produce those enzymes on the inside of the cell, and that enzyme is going to spend its entire life, if you like, on the inside of that cell, doing work and producing products on the inside of that cell, and that's what it's for. So the vast majority of them are going to be found inside our cell doing work for the inside of the cell. Whereas there are other types of enzymes that are released on the outside of the cell. So the cell will produce it on the inside. It will be released outside of the cell, where it will go out into the environment and do something. Now, a lot of the times, these particular enzymes are meant to digest things, to acquire nutrients. So in case of cellulase, it breaks down cellulose, or amylase, it breaks down starch. And then these organisms, the little bacteria, can suck up that digested, broken down nutrients and use it on the inside of their cells. Or in other cases, in this case, penicillinase is a type of enzyme that's produced by some bacteria that's released that breaks down penicillin. And this is how bacteria become immune to particular drugs. So they release these enzymes that go out, and the enzymes will tear apart the penicillin molecule so it can't actually harm the bacteria because there's none there by the time the bacteria gets to it. So that's pretty cool. And then the other times they can break down our skin or dead organisms and things like that, flesh, that are not particularly pleasant. So some diseases are caused that way, or damage, I should say, and things like that. So endo and exoenzymes, where they're used and where they're released. Here's some more enzymes, what they do, things like that. Take a look at them if you want to. So there's also two types of enzymes in reference of how often a bacteria produces these things, when they're present, how often they're present, and when and why we make them, or bacteria make them, as an organism. So the first type is called a constitutive enzyme, and this is something that's going to be present all the time. It's the constants part. It's going to be something that an organism is going to be producing, an enzyme that breaks down a very, very common nutrient source, or something that organism comes in contact with a lot. So it makes sense that if you come in contact with something a lot that you eat, that you make a lot of enzymes that break that thing down. So when you come in contact with it, which you more than likely will, you can break it down. You don't want to be missing that opportunity. So these types of enzymes are going to be produced all the time. They're always there, and in tons and tons of amounts, regardless of if the substrate that they break down, that nutrient source or whatever it happens to be, is there or not. So once again, kind of a hedging your bets sort of thing. You're probably going to run into that nutrient source, so it kind of makes sense to waste a little bit of energy to make enzymes that you're probably going to use, because you'll probably run into that nutrient source kind of thing. And then you have the other type of enzyme, which is called a regulated enzyme. And these are going to be either induced, which means we turn on the production of an enzyme, or repressed, which means we turn off the production of the enzyme. Now, these are going to come into play when things that aren't necessarily so common in the diets of bacteria and different organisms and things like that, but bacteria in particular, that we'll talk about for this one, when they come in contact with something that they don't necessarily eat all the time. So in your intestines, there's a lot of bacteria, E. coli, that break down lactose. Now, lactose is a milk sugar, you probably knew that one, that is not going to be a part of everyone's diet, or if it is a part of your diet, you probably don't eat dairy products every single day. So the bacteria inside of your intestines, when they come in contact with lactose, it's got a lot of energy in it, so to be able to break that down is a beneficial thing. But it's kind of silly for them to make enzymes all the time, lactase, that can break down lactose. It's just wasteful. You don't have lactose in your stomach all the time. Some people don't even eat dairy products, so they would just be wasting all this energy, wasting all this protein, building blocks and stuff to make it. So it's just kind of wasteful. So what happens is when you drink a dairy product or something like that, eat dairy product, the lactose inside of your stomach, inside of your intestines level goes up. The bacteria detect that. They start to sense the presence of lactose. And then they turn on the production of lactase, that enzyme that breaks down lactose. So they only turn it on when they need it. And then once they break down all the lactose that you've eaten, or you don't eat any more dairy for a little while, they will turn off the production of that lactase because they don't need it anymore. The lactose is gone in the environment. It's not there anymore. Why do I need to keep producing this when it's gone? So that's regulated enzymes. They're produced when when you come in contact with something that you don't necessarily always come in contact with, but it's nice to be able to eat or destroy, or antibiotics and things. The antibiotic resistance work this way. Bacteria don't produce those penicillinases all the time unless it's there, things like that. And then you got the constitutive enzymes, which are constantly there for breaking down nutrient sources that the bacteria eats all the time, or keeping the cell alive, things that the cell needs to do, like producing DNA and RNA and things like that. So the two different types of enzymes in that sense. So this is where the metabolism part kind of kicks in a little bit, when we're talking about the different types of chemical reactions and how enzymes do this. So there's basically two different types of chemical reactions. You have a synthesis reaction, condensation reaction, or hydrolysis reaction. So synthesis means putting things together. So taking two different pieces of something that are separate and sticking them together. And this is what enzymes are great at. Enzyme holds one piece, the other part of the enzyme holds the other piece, and then a little bit of energy is input, and those two things stick together. Now that enzyme is going to hold those two things, put them really close together, and do most of the work of getting them close together, and then just a little bit of energy has to be put in to bond them Whereas instead of the other way, you'd cell would have to go get them, bring them all together, and force them together, which would take a lot of energy. So the enzyme cuts out the kind of the middleman and saves us some energy. Now, when these types of bonds are formed, you can see here that a water molecule is formed. That's the condensation part here. We form water. This is what you breathe out. Water vapor and things like that produced by metabolism and stuff like that. And then you have our hydrolysis reactions, which are going to break things apart. You take two things that are put together and the cell wants to strip them apart, either to get the energy out or break down to smaller building blocks or things like that to use to build something else. So that's going to require the input of water to do that. So a water molecule is going to come in, provide the little bit of energy that's needed, and then the cell is going to break apart. The enzyme is going to break apart those bonds, and that water molecule is going to bond out and fill up that shell for those particular molecules. So it's going to strip it apart. So enzymes, you probably have gotten the sense already that the enzymes change. Cells have some that are constantly there, some constantly produced when stuff's there, some's turned off when things aren't there, things like that. They're different. But the activity of an enzyme overall, how fast it works, doesn't change. An enzyme has a set rate that it's going to function at. It can't get any faster, it can't get any slower. And that's just, it's kind of like your car has a speedometer and it's either at 100 miles an hour or off. So the second you turn it on, it just instantly jumps to 100 miles an hour kind of thing. So that's how enzymes work. But you can manipulate that, I should say they work at that rate at happy cells, at a normal happy cell. If your cell has everything it needs, the bacteria, for instance, if we give them all of their nutrients on a petri dish to have a happy environment and things like that, the enzymes inside of them will function as they should. They'll work at that rate. That doesn't change. But you can manipulate that rate by changing the environmental conditions that cell or those enzymes are held under as well. So if you heat it up or if you cool it down, if you increase the oxygen content, if you change the pH, anything that you do to that organism is going to change the cell, which in turn is going to change the enzymes inside, in the sense of if it gets too hot, you can break apart the chemical bonds. Or if it gets just a little warmer than it should, sometimes the enzymes work faster than it should, which is kind of nice sometimes. And then if it gets colder, they work slower. If it gets too cold, they stop working. So all of those different types of conditions, or pH can break apart bonds and things, are going to affect how a cell or a bacteria, for instance, for a single cell, produces energy and can go about carrying on its daily activities. So if it's not in a happy environment, it's either going to have to work harder, it's going to have to work harder to maintain life. So under a happy condition, it's got exactly what it needs. Its enzymes are working at this perfect rate. The amount of energy is what's been evolved for that cell to maintain. It should be able to maintain that and live happily and healthy. But if it's too hot or too salty or something like that, the organism is going to have to spend a lot of energy making more enzymes, because the enzymes might not necessarily be able to function as well as they should, or they might break down quicker in the presence of an acid or something like that. Or if they are working too fast, it's going to have to take more energy to power those reactions and things. So the cell, when it's in a non-happy condition, works a lot harder, which is bad for the cell. And that's why those conditions are so important for cells. Anyway, if you break apart an enzyme with things like acids or things like that, it's called denaturation. And this is when the shape of the enzyme, the protein portion of the enzyme, the bonds are broken by a heat or acid or something like that, and it falls apart. Once it's fallen apart, it's not going to be able to be put back together and function properly ever again for the most part. So once a cell has had its proteins denatured, it's pretty much going to die. And this is how a lot of our things that we use for detergents in our washing machines work. They denature the enzymes inside of the cells, and that's what kills them. They also break down the cell membranes and things like that, so that's what works on killing the cells. So cool on that one. So enzymes are really important for one another. in the sense of metabolism, because what happens is they play off one another. So what happens a lot is enzyme A will produce a product that's going to be used by enzyme B to make substrate C, which is going to be used by enzyme C to make substrate D. And they work in all these big circles to produce things. So sometimes we can turn off steps in those little circles, if you like, metabolic pathways. Or cells can turn off different steps in those pathways if they like to, via things called competitions or inhibition, if you like. And there's two different forms of inhibition. We have competitive inhibition and non-competitive inhibition. So competitive inhibition is going to be when something literally gets inside of the enzyme's active site and plugs it up. So this would be like somebody sticking a tube of super glue in your front door and squeezing, and then you can't get your key inside the door. So no matter how much the key fits the door, it's never going to be able to go inside, which means the reaction is never going to be able to occur, which means you're not going to be able to unlock the lock, which means the enzyme won't be able to do anything to break apart because it's blocked up. And then you have non-competitive inhibition. And this is essentially the lock and the key concept, where with the cofactor, you have to have the key before you can turn the lock. Where somebody's gummed up the lock. You put the key in, but the lock's not going to turn because the lock's gummed up. So this is how non-competitive inhibition works. It's also called allosteric, bonding in different sites. So essentially what happens is somebody stuck a door wedge, I guess if you like, behind the door. And you can put the key in, you can turn all you want to. It's just never going to work because somebody stuck a door jam behind it. So that's kind of how that one works. So different types of inhibition, and cells can use these for different types of things. Once again, if we don't want to break things down or we need to turn things on, we can do so. And we can also take advantage of this as humans by drugs. So we can turn off particular things that bacteria do to kill them using different types of drugs that function in this way, although it's not super common because we have enzymes too, and they tend to have some very fun crossover side effects. So you stop the production of the enzyme, you usually stop the production of the protein, which is going to stop the cell from being able to do things. So pretty easy on that one. So here's what enzymes do. Take a look at that one if you want to. Okay, so why are we doing all this stuff? Why are enzymes important? Why do we need enzymes to break things down, to make things for us? And the answer to that is the pursuit of energy. That's the answer to every question around life, to get energy. And energy is the capacity to do work or change a chemical substance from one shape to another. So work can be simple, lifting up this mouse, or in the sense of metabolism, putting a phosphate group onto an ADP to turn it into ATP, or stripping apart the ADP or the ATP to get ATP again to release the energy inside. So those different things are all different types of energy, the ability to do something or cause change in something. And there's lots of different types of energy. You've probably heard of them: thermal energy, heat energy that we use. You can observe that coming off the sidewalk, the heat of the coming up. You have radiant energy, is kind of the same thing. Electrical energy, familiar with that, turn on your light switch. Mechanical energy is the pushing concept, powering your bike and things like that with pedals. Atomic energy, chemical energy, ATP in that sense. So lots of different forms of energy, but all of those different types of energy do work. They all power something to do something. So different forms of energy exist. So there's basically two different types of chemical reactions that occur inside of a cell. You've probably heard this over and over and over again. And it turns to the different types of ways that cells use or release energy inside of those particular reactions. So you have an endergonic and an exergonic reaction. Now this is just a chemistry concept. It doesn't necessarily apply just to biology. There are chemistry reactions that occur the same way. But an endergonic reaction is a reaction that requires the input of energy. So this is when cells are going to build things up. So you're going to have one piece come into an enzyme, substrate A, substrate B. The energy input is going to come into the enzyme, and that's going to crack the enzyme together and force these two things to bond. And that's going to be our endergonic reaction. It takes energy to do that. And the other side of that, we have an exergonic reaction, which is a reaction that's going to release energy. Now, this exergonic reaction, when we build things up, they store a lot of energy inside of them. Nuclear energy, for instance, radioactive things have tons of energy stored inside of them. ATP, the bond, is very unstable. It holds a lot of energy inside of it. And the second that reaction breaks, the energy that's stored inside of it, or that bond breaks, I should say, the energy that's stored inside is going to be instantly released to the environment. Now, you can release that energy onto an enzyme, which is how endergonic reactions work. That ATP comes up, releases the enzyme as it breaks apart, releases the energy into the enzyme, which is what powers the enzyme. Or in the case of something like radioactivity, we have an explosion, or radioactive neutrons that float away. TNT, explosives, things that have lots of energy stored inside those bonds. And when you release it all at once, that energy is going to have problems causing explosions, or in this case, for bacteria, we can use that energy to power enzymes to do things. So two different types of reactions that occur inside of cells to break things down using energy, or to break things down and get the energy out, or to build things up, and that's taking energy to do so. Now, these things are really kind of interconnected with one another. They both kind of play off one another. So it gets a little confusing. So make sure that you just kind of keep apart. Endergonic consumes energy, endo putting energy in, exo releases energy, exo exit out, kind of thing. So cool on that. Redox reactions, these are the concept of transferring electrons around. I'm not going to be super focused on redox reactions for this one. So ATP. Now, this is our big one here. When we're talking about metabolism, this is metabolism pretty much in a nutshell. ATP, this is what we're going after. This is cellular money. Cellular gasoline is probably a better way to put that. It powers the cell. Now, it's a very simple little molecule. It's got an adenine group here, ribose, and then 3 phosphate groups bonded to that ribose sugar. Now, each one of these little phosphate groups, you start out with one phosphate group. You have a monophosphate, adenosine monophosphate, AMP, adenosine diphosphate, ADP, and then ATP, adenosine triphosphate, 1, 2, 3 phosphates. Now, each one of these bonds after monophosphate, this first phosphate bond and this phosphate bond here, hold energy inside of them. The energy that's going to be used to put them together is going to be stored inside of that bond. It's going to be sitting there, and it's going to be very high energy level. And the second that bond breaks, whatever energy that's stored inside is going to be released to do work with. So ATP is what the cell uses to power reactions inside of the cell, to make its enzymes work, to do things. Now, ATP is a very short-lived molecule. It doesn't survive in the environment at all. So the cell constantly has to make this all the time, or else it's going to run out of gas pretty much instantly and die, which is why you kind of have to breathe all the time so you get some more oxygen to make more ATP. So ATP production is a really important thing, and that's essentially what most of the cell is built about, is making energy. So this is a really important concept for cells, producing ATP, which is essentially what metabolism is. So once again, these two bonds here, ADP and ATP, the two bonds that hold those phosphate groups together, hold tons of energy inside of them. Kind of like the rubber band. The second the rubber band breaks, you twist it up, it'll spin out and release all the energy inside of it. It's essentially how this works. So ATP is made from ADP, essentially. So what's going to happen is ADP is going to have 2 groups on it. It's going to require a little bit of input put in from, and then we stick another phosphate group on it. So I'm going to kind of do some microbiology magic a little bit on this, kind of give a very simplistic overview of metabolism in a nutshell. So making ATP is going to occur by 3 different ways. Substrate level phosphorylation, oxidative phosphorylation, and photophosphorylation. Most of them you're familiar with. For the first one is going to be regular traditional metabolism, pretty much. Oxidative phosphorylation is a little different. And then photophosphorylation is things like plants or cyanobacteria using the sun to do this. For us, we're going to focus pretty much on the traditional metabolism, three-part glycolysis, Krebs cycle, and the respiratory chain, things like that. So as you're probably aware from our previous lecture, bacteria live in tons of different environments, places that have lots of oxygen, places that don't. So the way that they break down their food and get the energy out of it can't necessarily always be dependent on oxygen. So you can't do the traditional sense of metabolism using oxygen as your electron transport chain final acceptor if you don't have oxygen present, or you can't survive in the presence of oxygen. So organisms that just carry out traditional metabolism and use oxygen as their electron transport chain final acceptor undergo something called aerobic respiration, breathing in the air. And then you have the opposite of that, where organisms use something other than oxygen, and this is anaerobic respiration. And then you got fermentation, which is a very simplistic form, a kind of a last-ditch effort. We're running out of food. This environment's not so great. Form of making energy using just glycolysis. So a very simplistic form, doesn't make a lot of energy way to do this. So different types of nutrients, different environmental conditions are going to influence when and how these are used in different bacteria species and things like that. So different ways to produce the same thing. ATP is essentially what all this goes for. So these are the different pathways that are involved with this one. Glycolysis, the Krebs cycle, the electron transport chain, using oxygen as our final electron transport acceptor. And then you get 38 on average. Somewhere it's a lot lower than that because it takes energy to make energy, and it's not always the most efficient thing on the planet. In theory, you get about 38 or so ATP out of this entire process. Anaerobic respiration, same concept, uses something other than oxygen. You can use these presence of things to help identify different species of bacteria, which is cool. And then different bacteria produce more or less ATP depending on their species. And then once again, glycolysis, fermentation, very last ditch, using only glycolysis effort of making energy using fermentation. And you're going to produce, this is where we get our alcohols from yeast and things like that, or acids and stuff from bacteria and things, vinegar and things like that, yogurt. So you get about 2 ATP from this, so not a lot of energy. You kind of a last-ditch, desperate attempt to not die and make some energy kind of thing. So aerobic respiration uses the traditional glycolysis, electron transport chain, Krebs cycle, things like that. It's also called the carbon cycle, depending on the books that you're in and things like that. It changes around from time to time. So essentially what this is going to do is it's going to make ATP. Very little of it. At the beginning of it, you're going to get 2 out of glycolysis. You get 4. It takes 2 to start the whole thing. It takes energy to make energy. So you put 2 in, you get 4 out. 4 in, you get 2 out. You put 2 in, you get 4 out, so you make 2. Krebs cycle makes a little bit, and then the electron transport chain down here is going to make a bunch. You've probably seen this concept before. I'm going to post a video of how this thing works in detail if you want to check that out. Watching the little electron transport chain, the ATP synthesis thing spin around and form the ADP, and the ATP putting the energy in and things like that. So that's kind of cool. Glycolysis, here's the steps of glycolysis. Big, long process to essentially take one glucose molecule and split it into 2 things called pyruvic acid. You got one glucose, it's got 2 sets of 3, so 6 carbons stuck together. 1 6-carbon glucose. Takes a little bit of energy, we put in some ATP. Break that ATP bond down into ADP, the energy in ATP is transferred to break apart, phosphorylate our glucose molecule. Then we're going to change it further, we're going to break it down with a little more energy. Then we're going to finally split it from a 6-carbon molecule into 2 3-carbon molecules. And then we got to change those 2 3-carbon molecules around again to get them to what we need. So we're going to have to change those 2 3-carbon molecules again into 2 3-carbon molecules, eventually called pyruvic acid. And that's essentially what glycolysis is going to do. It's going to take glucose, split it in half into 2 pyruvic acid molecules. Those pyruvic acids are going to be needed to start either the Krebs cycle or fermentation, depending on what you're going to do with them. So pyruvate's going to do different things depending on what pathway it ends up in after it's done. So it's either going to go out of glycolysis, and it's either going to end up in an anabolic pathway, where it's going to be sent to make amino acid, sugars, things like that, or it's going to end up in the fermentation pathways and things like that for yeast, where it'll end up making alcohols, or bacteria, acids and gases and things like that. So pyruvate's going to do different things depending on which method of making energy the particular organism employs. Now, the Krebs cycle, really long pathway to get out the other end of it. So essentially what we're going to want is this guy right here, NADH. And that's our big guy right there. So we kind of want that guy. We're going to make a little bit of ATP on the way, not a whole lot, but we kind of want that NADH out the other end. So that's what we're looking for. So essentially what's going to happen is we're going to use NADH and another fun molecule called FADH2 to kind of move and shuffle around electrons down the electron transport chain to make tons and tons and tons of ATP via oxidative phosphorylation. Now I'm going to post a video carrying you guys through this in a little greater detail. It's kind of difficult to go through this one asynchronously. So I'm going to post a video going through this in a little more detail and a lot more animations, if you like, that kind of stuff. So the electron transport chain, it takes place inside of a mitochondria, inside of eukaryotes, or on the cell membrane of bacteria. And it's going to pump the hydrogen ions into the periplasmic space, or in the outer membrane or inner membrane space of the mitochondria. And essentially what's going to happen is the NADH and FADH are going to funnel the hydrogens, strip the hydrogen off back down to NAD and FAD, strip the hydrogen ions off. Those hydrogen ions are essentially going to move around inside of the inner membrane, and then they're going to be funneled down very quickly through this ATP synthase thing here. So what's going to happen is you have ADP, we want to make it into ATP. So we take ADP on one side, this is the enzyme part, ATDP over here, the P over here, the phosphate group. We're going to make those two things stick together. Hydrogen's going to shoot down here real fast. I'm going to provide the energy needed to make this enzyme work. And when it does so, it's going to make this little enzyme kind of spin like a boat motor, forcing the ADP and the ATP to bond together. So that's how we make ATP. So that's why we need lots of hydrogens and things like that. And then that hydrogen ion is going to be essentially gotten rid of via water, an oxygen molecule to make water. So that's why you have to breathe. So if you don't have oxygen, you don't have, your hydrogen ions back up. They don't go away. They back up. There's too many of them. Things like that. They overload the system, and you die. So that's why breathing's so important. So this is essentially how bacteria do it. It all takes place in their cell membrane between the cell wall and the periplasmic space. It's essentially the same kind of concept, just a little more simplified. And they use the same spinning little ATP synthesis concept here, just a little more simplified version. So in theory, we get 38 ATP, or 36, somewhere around in there, and about 6 water molecules. So if you do that, you get water vapor, and that's what that is. So you can see here that each step, glycolysis and things like that, Krebs cycle, or electron transport steps down here, what is going to come out of each step, what's put in, and then what comes out. So really the only things of super importance are pyruvic acid, the NADHs. Those are the things that are going to be moving molecules around. So all these H's, the hydrogens, are going to essentially be stripped off at the end to power the electron transport chain, and then we'll stick the hydrogens on later again to recycle the FAD into FADH into NADHs again and stuff. So the hydrogen stripped off, the NAD goes back, picks up another hydrogen somewhere else, and starts the process over. So essentially how that works. Anaerobic respiration. Now in this case, we still got to make energy, we just can't use oxygen. So they're going to use something else to strip away those hydrogen ions at the end. And that's essentially how that works. In most cases, it's some sort of nitrate or something like that. That they're going to use to get away those hydrogen molecules and things like that, something other than oxygen. So fermentation, once again, is that last kind of ditch effort to not die. Very little bit of ATP is produced, but once again, we do get some good things from it. If you like alcohol, if you like yogurts and cheeses and things like that, got to get a lot of good stuff from that products of that as they produce energy via that particular pathway. Here's a kind of a table kind of comparing the two if you want to take a look at that. Now, most of these metabolic pathways, breaking things down, building things up, work in both directions. And this makes perfect sense if you think about it. So sometimes a cell needs to, build up its cell membrane. So it can do that by taking lipids from the environment, breaking them apart, and putting them into the cell membrane. Now, sometimes the cell is going to get into a not particularly nice place, and it can be running a little low on fatty acids and lipids and things like that, so it can tear apart its cell membranes and use those lipids for something else. So it can use them to make energy, and that's what happens when people or animals start to starve to death. The body starts to tear apart its own particular things that it's built already. So we build things up, but we also tear them down. So metabolism works to build up the cell membrane, but it can also easily strip apart the cell membrane and eat it for food if the environment goes bad or the organism's not properly fed or something like that. So most of these metabolic pathways, even though they mostly build things up, will easily strip them down just as well for the energy that they put in to start with. It's kind of like a savings account. You don't intend to pull the money out, but if you need to, you will. And that's kind of how this works. So these are referred to as bidirectional or amphibolic, which means they go in both directions. They can build them down, build them up, but they can instantly tear them down if they need to for the energy and building blocks stored inside. So you can see here what the particular things that we build, amino acids and things like that, can easily be stripped apart to pull out the H2Os, the things that we need to make amino acids, to make carbohydrates and make fats. We can instantly just start ripping apart our own cells in amino acids and things to get that energy out. So then photosynthesis, everybody's favorite. The production of energy via light. This is super cool. Using oxygen, or CO2, and water to produce glucose and oxygen. So in this case, no ATP is going to be produced yet. The glucose will then be stripped apart, sent through the traditional processes to make energy and ATP via that way. But these guys make glucose, which is super cool. They got to make the gasoline, the ATP, but they got to have fuel to make gas with. So these guys make the fuel to make the gas. So this is super neat, or the precursors to make the gas, if you like, the glucose. They make all the original glucose, plants and things. So it comes from the sun and raw products that are floating around in the environment, such as water and CO2. So super neat. There are two different reaction stages that occur in photosynthesis. One of them is light dependent, it needs sunlight, and the other one can occur in the dark. And that makes sense if you think about it, because plants don't always live in the sunlight. It would be really kind of a counterintuitive, especially for plants. There are also bacteria that do this as well. And it would not make a lot of sense for an organism that lives 24 hours a day to only be able to make energy in the half of the day that you get sunlight. So it's kind of silly. So what they do is they make a lot of product during the daytime when they got sun, and then they store it, and then they'll tear it apart later when the sun's gone. So they can do stuff during both times. So that's kind of neat. So you get the light-dependent reactions and the light-independent reactions. So those are the easy ones on that one. So the other stage of photosynthesis is referred to as the dark reactions, or the Calvin cycle. If you some books. So the light-independent reactions are sometimes called the dark reactions, or sometimes called the Calvin cycle, and they're going to essentially take CO2 that they can. A plant doesn't have to have sunlight to get CO2; they can just breathe in CO2 outside in the environment all day long, into this little precursor thing called ribulose bisphosphate, or Rubisco. Rubisco is kind of the precursor to eventually make glucose. Rubisco is one of the worst molecules on the planet. It literally has about a 0.2 second shelf life, It takes a ton of them to make anything out of it. It was just one of the very first enzymes to evolve. It worked, and that's what it ran with, but it's extremely inefficient. It's also it is the most numerous enzyme on the planet by weight, because plants have to have more of that than anything else to power photosynthesis, because they have to constantly make it, because it falls apart the second that they make it, or it's broken down. That ribulose bisphosphate, not a very useful molecule. Works really well for what we need it to, but it could work so much better. Here's the reactions of photosynthesis. Essentially, the very end of it is the electron transport chain. Works kind of pretty much the same way. Here's the reactions of the Calvin cycle. Essentially, the exact same thing. We're going to break apart ribisco into smaller molecules that are eventually going to turn into glucose. Pretty easy on that one. And that's pretty much it for metabolism. I'm going to post a couple videos to give you guys a little more in-depth view of this if you'd like it. A little dig deeper to watch these things actually move around. But other than that, if you've got any questions on this, or if you want me to go a little deeper in anything, just let me know, and I'll be more than happy to. Other than that, guys, have a great